I hope by the end of this, you'll have a better intuitive sense of what LDL and HDL really are. So if you look at food like this, one reason why everything here is so highly recommended in your diet is because there's actually very little fat, very little lipids in which you see in here. And the question in diet advice for years has just been to be how much fat is good for you and how much is bad for you. And so what we're going to do today is look at the question of what is lipid really, and how is it handled and digested and used as an energy source in our bodies. So first of all, let me say that in chemical terms, unlike sugars and unlike proteins, where those words define a particular type of molecular structure, with the lipids, the terminology is more ambiguous in that lipids are really solubilized primarily as molecules that are not soluble in water. And some of them, as we will see, have very different kind of molecular structures. And of course, they come in various forms. So various kinds of cooking oil, olive oil, canola, etc., can be liquids. Others can have various degrees of hardness, and so lard, butter, cream cheese, etc. These are the way we encounter lipids in our food. For most lipids, there is a core structure that all of them contains, but not for all. So, I mean, to jump ahead a little bit, cholesterol, sterols are also lipids, but they don't have, they have a very different kind of structure, what we'll talk about. Most of the lipids that we encounter in our food contain these molecules called fatty acids. And so what is a fatty acid? And they come in this saturated and unsaturated form. So to a chemist, something that is fatty is like this is called a hydrocarbon. Now, it has only hydrogens and carbons in it, except for this N down here. And in fact, you clip off this COO2, and what you've got left is the same molecule, the exact same molecule that you find in gasoline. And its main characteristic is the total lack of oxygen. Now, it can be in the form in which every carbon is attached to the maximum number of hydrogens. That is fully saturated. It is full of hydrogens. On the other hand, this chain of carbons can have situations where this carbon, for example, in forming its 4 bonds, formed one to carbon, one to a hydrogen, and then formed a double bond to the other carbon. And that means that it doesn't have as many hydrogens as it possibly could. It is unsaturated in that it would be possible to add hydrogens to these other carbons. And in dietary advice, we often hear that unsaturated fatty acids are good for you, and having too much saturated fatty acids is bad for you. Now, let me say right now that this has always been a difficult thing to measure scientifically. And in recent years, this has gotten revised a little bit in that the badness of saturated fatty acids has been de-emphasized. And that saturated fatty acids and unsaturated are really not bad for us per se. They're good food. They're very high energy food. It's just like so many other things. If we get them in excess, that's when they start to be correlated with cardiovascular disease. Now, the principal chemistry that's important to know about fats is that this whole section of the molecule, which has no oxygen in it, has its electrons very evenly spread over its whole surface. So it really doesn't develop much plus or minus charge. And therefore, it it very weakly binds water. It basically doesn't bind water. So we talked about the important chemistry of oxygen in that it has a high affinity for electrons. And so in a water molecule itself, the electrons tend to stay around the water, and that makes it negatively charged a little bit. Whereas the hydrogens out here are kind of deprived of their electrons and they're slightly positively charged. Now, this molecule is called a fatty acid because this is the fatty part, and this is the acid part. There are oxygens out here. They can develop partial negative charges, and they will bind very readily to the plus and minus charges of water. So this chemistry, the fact that it binds a tiny bit of water on one end, but not on the other end, really is essential to understanding the role of it in our bodies. Now, what about saturated versus unsaturated? Why would these have any different effects on our health and on their chemical properties? Well, if we look at these molecules with a different way of representing how the atoms are put together, and we use one of these, what's called a space-filling model, that is, you represent all the atoms as spheres of the right size. If a fatty acid is fully saturated, it's a fairly rigid, straight chain molecule. So fatty acids differ not only in whether or not they have double bonds or not, but in the length of these chains. So they all have different names like palmitic acid or stearic acid. But the easy way to remember them is just with the number of carbons. So this is 2, 4, 6, 8, 10, 12, 14, 16. And it's always an even number. That's because when they're built 2 at a time. So this is a 16 carbon straight chain. This is an 18 carbon straight chain. Now, if you put in a double bond, if it's unsaturated, what that does is to cause a kink in the molecule. And this explains why oils, like olive oil, have a lot of unsaturated fatty acids in them, whereas solids, like butters, are primarily saturated fatty acids. You can see that if you took 20 of these molecules and lined them up, they could all lie next to each other and form a very complex solid. But if you put these in here, this kink is going to prevent the molecules from really lining up next to each other. And therefore, they're going to be liquidy at lower temperatures. All right. Now, there is a third type that you find rarely in nature, but it used to be a big issue in food. It's really not an issue in the United States any longer. So a lot of us certainly remember in our youth that it was very common to eat a lot of margarine in the diet. And the way that was produced by the food processors was to take plant oils, like soybean oil or canola, and those plant oils, being oily, not surprisingly, turned out to have a lot of these unsaturated fatty acids in them. Well, you could chemically add hydrogens to these bonds and make them fully saturated. And if you did that, then the oil would harden. And so you could take canola oil and make margarine out of it as a substitute for butter. However, the way the chemistry was done initially is once in a while, you ended up with molecules that look like this. You failed to fully add the hydrogens, and in fact, the hydrogens got flipped over. See, what happens here is the hydrogens are next to each other in the molecule. That's called the cis configuration. But here they are across, and that is the trans configuration. So this is the structure of a trans fatty acid. It was found that trans fatty acids were correlated pretty highly with the occurrence of various kinds of cardiovascular disease. And because of that, they've been pretty much outlawed in the United States. It's not as though we never encountered them in nature. There are 1 or 2% of the fatty acids in some food sources are trans fats, but we can deal with them. It was in the higher amounts that you found them in these saturated margarines that caused the medical problem. However, making trans fats in the way making saturated fats in food processing in a way that produces this trans bond, that is now illegal in the United States and in probably most of the developed world. But trans fats are still common because they tend to be cheap in large parts of the underdeveloped world. Okay. Now, what happens in our bodies is that much of our fat is in the presence of a true fat. What do I mean by that? A little bit of terminology here. So we tend in dietary language and food language to use the word lipids and fats interchangeably. And that's fine if you know what how your definition is. But to a chemist, a fat, in fact, is a particular chemical structure, and that structure is shown here. So a fat truly is a triacylglycerol. Now, what does that mean? So the 3 carbons we have up here form the molecule glycerol. And glycerol is glycerin. It's kind of a sweet molecule. Sometimes it can be added to some food. But if to each of these carbons, you attach a fatty acid. So remember, the acid had the 2 oxygens on it. And this linkage between the carbon and this fatty acid is called an acyl linkage. Then you get a molecule that has glycerol as the carbon skeleton here with 3 long chains attached to it. And these chains can be saturated or unsaturated. The carbons can be anywhere from 12 to 22 carbons long. But if you have all 3, a triform, 3 acyl bonds to a glycerol, then you get a space-filling model that looks like this. It's a very compact molecule. It's a very high energy molecule in that if you counted all the electrons in this molecule, there would be, probably well over 500 electrons, and you can use the energy in those electrons to make ATP. So in our food, we convert a lot of the lipids into these triacylglycerol molecules. They're water-free. They form big fat globules, if you will, and they are transported to a special storage tissues, our fat cells, and that is called our adipose tissue. So if you look at these cells, you can see they've actually been stained here. So you can see the outer membrane, but inside you don't see much structure at all. If you really got an electron microscope in there, you could find a few mitochondria and you'd find the DNA, et cetera. But mostly they are just bags of these triacylglycerol molecules. So if you think of what your fatty tissue looks like, this is it. There are just a few capillaries running through there to supply blood and nutrients to keep all of these cells alive. Because in hard times, you're going to want to use this. This is your storage material, and you have a mechanism to make it and store it, but you also have a mechanism to retrieve it and to burn it. We'll talk just a little bit about how that happens. Okay. So the other way that we have a lot of lipid in our cells, besides stored as fat, is we use lipid to form our cell membranes. This is why it is so important in our diet. We really must have lipids in our cells to survive. Now, the molecule that forms our membranes is a quite complex molecule. So once again, we see the 3 carbons that are in glycerol. And if you attach a fatty acid chain to 2 of those carbons, and that fatty acid could be saturated or unsaturated and of various lengths. But then to the 3rd carbon, you don't attach a fatty acid. Instead, you attach something that can bind to water molecules. And here's a typical one. It has a phosphate connected to a small molecule. I think this is a choline molecule, I think. There are several places on this molecule that can bind water. And if we put it in the space-filling model, it looks like this. So we have our 2 fatty acid chains down here that won't bind water, and we have this head group up here that will bind water. And you put a lot of these molecules together, and all of the parts that don't bind water will associate with each other. Now, it's not because they're repelled by the water or they don't like water. It's not that they don't like water. It's that water doesn't like them. And so they're excluded from the water and they just tend to hang out together. But the result is that we've got this layer of lipid that forms a good insulating barrier. It prevents small molecules that are dissolving water going from outside our cells and inside our cells. So it's really the boundary of our cells to the outside world is formed by these phospholipids. So we have a cartoon over here, what a typical biological membrane would look like, in which the core of it is these phospholipids with the lipid tails down here and the head groups up here. All right. And then there are proteins that float around in this lipid layer, and they allow molecules to be transported in and out. Yep, somebody was not unmuted there. Okay, now I'm gonna take a pause here because I've shown you all of these structures, and I want to just give you an opportunity to ask questions. Anybody have a question? You can unmute yourself about any of this structure. Fats, and these are called phospholipids. Any questions about this?
Hi, this is Helene. Just to be clear, the saturated fats are not just straight bad, but it's really the trans fats that are artificial, industrially made, that are bad for our hearts?
That's right. So trans fats are present at very low quantities in our regular food, in plants and animals. We just don't make them. And so we don't have then the enzymes to deal with them and to degrade them in our cells because they don't occur naturally at any significant level. Now, saturated and unsaturated, we need both. We make both. The problem really comes about if you get an excess, especially an excess of the saturated fats. And that is, it's complicated, but at its core, it's associated with the fact that the saturated ones are so insoluble in water. And so, like when you get plaque formation inside your blood vessels, that can be driven by an excess of saturated fats. All right. But otherwise, basically you have to have them. And it's okay to eat a fair amount of them. It's just a caloric excess that causes the problem with saturated fat. Okay, now I want to show you a little demonstration here. I tell students the beauty of the formation of these phospholipid bilayers. How do we assemble them? Well,
there was another question.
Lady go ahead.
Harriet, she wanted to ask a question.
Okay, good. Thanks for letting me know. Go ahead.
Well, you might come to this. in some discussions that I've had with people about the omega-3 fatty acid,
Mm-hmm.
they will say that it's better to get it derived from fish. And if you concentrate, if you get them from vegetables, there's a benefit to fish when you compare it, because the fish have long chain molecules or whatever.
Yeah.
And I don't know what's true about that. And I don't know what advantage a long chain versus whatever other chain there is.
Yeah, I'm going to address that explicitly, Harriet, in just
Okay.
a bit. Okay. All right. So the beauty of this is that these phospholipid bilayers can assemble spontaneously. And when teaching this in my biochemistry classes, I decided to put together a little instructional video that shows how, in fact, you can demonstrate the spontaneous assembly of these lipid bilayers. So here we go. It's about a 5-minute video. Okay, I want to give you a demonstration of how lipids can spontaneously form bilayers in water. So what I have here is some water that has a little bit of ether in it. Now the thing about ether you have to be careful with because it's highly flammable. And in fact, we're going to test whether in fact a little bit of ether dissolved in this water is indeed really flammable. Okay, I'm going to fill this petri dish right up to the very tippy top. All right. And if it's really flammable as advertised, we should get a little, whoa, all right. It is flammable. Okay. So we could put out this flame if we could form a lipid bilayer in the top of that water. So we need some lipids. Easy source, your ear. Just touch your ear slightly to there. Put it right on the surface of the water. What you get is a form of lipid that will form on the surface. It will rush across the surface of the liquid and put out the flame, because there's now a very thin layer of lipid molecules on it. Now, maybe it was a trick. Maybe I put in some retardant or something. Let's give it a test here. It should not stay lit. Little flash, but it won't stay lit because of the lipid layer. All right, now, if it's really a lipid layer there, I ought to be able to get a glass rod and pull that lipid layer back. And that's what I'm going to try and do. So I touch it right to the edge and I pull the lipid layer back, right? Get the matches there. And I'm going to have Rusty go ahead and see if in fact, go ahead and light that side and see if in fact the layer is pulled back. Whoa, okay, that really worked. All right, now, if there's really a lipid layer there, I ought to be able to lift up the glass rod. It runs back across the surface of the plate, reforms, and puts out the fire. Okay. And so this is a really nice demonstration that in fact just a tiny amount of lipid will spontaneously form an impermeable layer in a water lipid interface.
Barry, I just didn't get, where did that lipid come from?
What? Well, on your surface, especially in your ear, you have earwax, and that's lipid. And so just a very thin layer, you don't even have to, enough that you could just feel it, will form just a single molecule layer to form this fire barrier. I saw this done in a seminar years before, and I've been copied it in my classes ever since. Yeah. All right. So now we're going to get to Harriet's question. So there's another group of lipids that are called the essential fatty acids. Back when I gave the first talk and I gave the list of 50 molecules that we absolutely have to have in our diet to survive, there were 2 of them that were fats. And these are this alpha-linolenic acid and linoleic acid. And these are the fatty acids called omega-3 and omega-6 that you see references to for dietary recommendations. Now, the terminology here is that the Greek letters starts with the alpha and omega. So the alpha carbon is up here and the omega carbon is at the end. And if you count carbon number 1, 2, 3, if at the third position you have a double bond, then that is an omega-3. And down here you got 1, 2, 3, 4, 5, 6. If at the sixth position, then you've got an omega-6. And we do not have the enzymes to put these double bonds out at the far end of the fatty acid molecules that we make in our bodies. We can synthesize all the rest, but for whatever reason, it's not just humans, it's missing in all mammals. Mammals have lost the ability to put in these double bonds at this end position in the hydrocarbon chain. And yet we need these molecules to make some essential signaling elements in our cells. And I'll tell you what these are. So if we look at the structure of these essential fatty acids, so we can compare an fully saturated molecule with 18 carbons. 18O means 18 carbons, 0 double bonds, stearic acid, or a mono unsaturated. Mono means 1 double bond. So this one is 18,1. But the double bond occurs not at 3 from the end or 6 from the end, it occurs 9 from the end. We can synthesize this in our body. All right, or we can have these trans ones that we talked about. But the essential ones are like this, linoleic acid. And what's interesting is they have a pair of double bonds, and so they make something that's almost like a hairpin. The molecule comes almost but not quite back. And it's this unusual structure that the biochemistry exploits to use this as signaling for certain processes, inflammation being a key one. All right, so now this is a little complicated because these are big molecules that go through multiple steps to get processed. But what we need in our diet are these two. We need the omega-6 one, and we need the omega-3 one. All right, and we can get this from some plant sources. However, if we eat these, then we can convert them to more complicated, longer chain fatty acids. So if you remember your Greek, icoso is something with 20 carbons in the chain, and docosa is something with 22 carbons. Now, the generalization here is that for the omega-6, we can get this from a variety of plant sources, and in your body, we can make the longer chain one that ultimately ends up in the signaling molecule that we need. All right. In a very rough sense, this molecule here, long chain fatty acid, is needed to stimulate inflammation within our body. And at times this is needed in an important physiological event. The complication is really more with the omega-3 side, in that we can get this in our diet, but the cells in our body are very inefficient in converting this to the 20 carbon form. And so that's why there's this recommendation to get this molecule, not in its precursor, but in the completed form. And one of the best sources for that is fish oil, and especially oily fish. So like, 1 ounce of mackerel is equivalent to like 6 ounces of catfish. The more oily the fish, the better in terms of the content of these essential fatty acids. And then this one, once is synthesized, our body can easily convert to the final signaling molecule. So this is an issue, for example, especially if you're really strict vegan or vegetarian, is that although you can get some of these, you can get it from flaxseed, for example, most of this gets degraded in your intestinal system, and of the part that is taken up into your body is inefficient in converting this making this 20 carbon form. And the nutritional people find that people who are vegans often have somewhat low levels, not necessarily suboptimal, but borderline levels of this. Now, where do the fish get it from? The fish don't make it, it turns out. They get it from eating, the big fish eat little fish that eat certain kinds of marine algae. And they know what these algae are. And there are these algal oils made that are rich in this substance, and they can be a good food supplement. If you don't want to eat fish, you can go directly back to the original oil and get it. Now, why are these important? As I said, the biochemistry here gets really complicated. There are multiple pathways that branch out, and I don't think I'm exaggerating to say there are 20 different kinds of signaling molecules, but what they do is really they ramp up or ramp down the inflammatory response. If you get a wound, what happens is there's a signal, and these molecules are part of that signal that say, bring in blood cells to this area because we have to repair this wound. And there are other signals that say the body is overreacting. We have to tamp down this regulatory response. And so it is these signaling molecules made from these essential amino acids where all of this biochemistry comes into play. Okay, so those are the essential amino acids. Now, let me turn to one other thing that's the kind of the health fad of the moment. There's always something. So the new thing being advertised as superfood in the last few years is coconut oil. So the question is coconut oil better for your health? So what is this all about? Well, let's compare coconut oil versus butter. Now, what we find is they're both high fat, high lipid foods. And so for a single serving size here, which is defining as 14 grams, first of all, we see that coconut oil is slightly more caloric than butter for a variety of reasons. But the major difference in it is the saturated fat. Coconut oil has higher saturated fat than butter does. Both of these actually have a little bit of the omega-6 fatty acids, all right? Now, the reason some of the health food people are pushing coconut oil is because they say, yes, it's saturated fat, but it's a different kind of saturated fat. Most specifically, that the chains are much shorter than you find in butter and animal sources of fat. So the typical unsaturated chains in butter might be 14 to 18 carbons long, whereas in coconut oil, it has a lot of chains that are 12 carbons long. There was an initial experiment done with, well, let's feed people a diet that is enriched in these short chain saturated fats. Now, in this study, they didn't use coconut oil, they used processed coconut oil, so it was much enriched for this. Because regular coconut oil is only about 15% of it is the short chain saturated fat. Most of it is the longer chains that you find in other kinds of molecules. You know, compare this with sunflower oil, which is mostly different because this is a fatty form here because of the presence of these unsaturated oil. So bottom line, I mean, the reading that I've done in this, first of all, they say the science in the clinical trials so far to show that there's a health benefit for coconut oil just doesn't support it. It's not very well studied, but there have been a few clinical trials. There's some evidence of some, one of the predictions is there'd be weight loss. It's the usual thing there. It tends to be very short term. But as far as any other effect, and what they're mostly measuring is whether this is changing your blood cholesterol levels, whether your HDL and LDL are changing beneficially. So far, the science is not really supporting an advantage for coconut oil. I'm going into this a little bit because the last couple of times I taught this course, this question came up, so I've done a little reading about it. I don't know, any question on the coconut oil business there? All right. Okay. So
I have a question.
yes, go ahead.
Sorry, just a quick thing. You mentioned butter. I've also heard alongside the arguments about coconut oil, people proclaiming the benefits of ghee, G-H-E-E, you know, the clarified butter that in the Indian diet, that there's something, it's not wholly butter. There's certain elements that are taken out, it's clarified, and it supposedly has, it doesn't have the same negative properties that regular, full butter does. Is that the same argument that as you've been describing with coconut oil?
I do not know anything about that. I hadn't heard that, and so I can't help you. I haven't read anything about that.
Okay, thank you.
I would say, you know, with butter, it's the same old story. Is butter bad for you? No, it's fine as long as it's not in excess.
But I was just looking something up here. It says ghee is jam-packed with conjugated linoleic acid, so a fatty acid associated with a long list of health benefits. So that's the claim. Does that make sense to you or not?
Well, that's one of the essential fatty acids. So I guess they're saying it's higher in the essential fatty acids.
Okay. Thank you.
I'll take a look at that. You have my curiosity out now. Okay, and I'll try to remember that for next time. Okay. All right. So reasons lipids are good for us is because they are such a powerful energy source. So if you look at this saturated fatty acid, we use some of these to build our biological membranes, but the main function of fat in our diet is as a source of energy. So we have enzymes that take them apart 2 carbons at a time. Really kind of an amazing complex set of enzymes. They sort of function like a little molecular wheel. One holds the end of the fatty acid chain as it's passed around to different enzymes and clips the carbons off 2 carbons at a time. And so this 18 carbon fatty acid will be converted into 9 2-carbon units, which are called acetyl groups. And these acetyl groups go into the same metabolic pathways we talked about with sugars. I'll show you right here. With sugar, we started with sugar here. We went through glycolysis, and ultimately we got a 3-carbon that then turned into a 2-carbon acetyl groups. So when you're digesting lipid molecules, just cut them into 2-carbon pieces. They go into here. This occurs inside the mitochondria. They go into this Krebs cycle reaction, generate lots and lots of NADH, that's that electron carrier, and that goes to power the synthesis of ATP. So I show here, just a single one of these molecules can result in the production of 108 ATPs. I mean, the power of lipids, one of my interests is I'm kind of a bird person, and I was just reading an article yesterday, in fact, on the records for bird migration. There are birds that can fly all the way across South America without stopping, 8,000 miles nonstop. How do they do that? And the answer is they are fat fueled. So they just load up fat, and so they get big layers of adipose tissue. And it's such a great way to store energy because it's so dehydrated. There's not a lot of water in there. And they fuel this directly into their mitochondria to generate all the ATP to make their muscles work to fly for 8 days. And brings up another interesting part. How do you fly for 8 days without taking a drink of water? And I didn't put that diagram in here, but when we looked at the metabolic pathway, where do those carbons and acetyl groups actually end up with? They get ultimately combined with oxygen to make a fair amount of H2O. This is called metabolic water, and flying birds use their fat not only to make energy, but all those hydrogens on that hydrocarbon get combined with oxygen to make water. And so they make their water while they're flying. Pretty amazing. All right. So let's turn to our kind of final important group of lipids here. And these are the sterols. So cholesterol is an important component of animal membranes. There are other related sterols that make up plant membranes. All biological membranes have somewhere from 10 to 15% of the lipids that form that bilayer, not being those phospholipids, but being these sterols. So you can see that structurally, they look very different. The main part of a cholesterol molecule is this 4 fused rings. Again, each intersection here is a carbon molecule. So here's a 6-carbon molecule, another 6, and this one is 5. And then attached to these 4 fused rings is this rather short hydrocarbon, CH2 chain. We make this molecule, we synthesize cholesterol in our bodies. Most of it is made in our liver. It is a very complicated system of chemical reactions. I think there's something like 26 different chemical reactions involved in making a molecule of cholesterol. We kind of start out with little 3-carbon pieces, and we put them together to make a ring, and then on that we build the whole bigger ring, and then we do these little methyl group additions here, and we add the hydrocarbon tail, and finally we end up with a cholesterol molecule. Now, space filling model shows that it looks like this, and here it looks a little bit more like a phospholipid in that you have a small group on the end here that will bind water. See, there's an oxygen there that can be partially charged, that will bind water. But all the rest does not bind water, and it's a kind of lumpy kind of lipid-like molecule. Now, as I said, this is essential in our bodies, but it's also been long known that in cardiovascular disease, we get accumulations, plaques forming in our blood vessels if we have high levels of this cholesterol circulating in our blood. And one of the most widely used drugs in the US and the world are these statins that are able to block the synthesis of this cholesterol. So let's just take a look at the biochemistry involved in here. How do we use them normally, and how do we deal with the medical problems that come from cholesterol excess? All right, so we eat a hamburger. That has a lot of stored fat, partly that has a lot of lipid in it. The lipid comes from the fat that's in the meat in the hamburger, and it comes from the special sauce, all of the oil that is in the hamburger. In our digestive system, we have enzymes that break those more complex lipid structures into simpler little structures, like the fatty acids, where we just have the water binding part on one end and the hydrocarbon chain. And there are transporters in these membranes that take them up into the cell. But then, unlike sugars, which are just passed through and put into the blood, these lipid molecules, because they don't bind water very well, have to be put in special packages. And so there are compartments in our body that gather all of these lipid molecules together and form these little lipid droplets, if you will, called chylomicrons. And then they are transported out of the cell and either go into our blood or into our lymphatic system. Now, how are they transported? This is where LDL and HDL come out. So I'm sure almost all of us, when we go to the doctor, he does our blood panel, and one of the things he wants to see is what are our various cholesterol levels. And so there will be numbers there for total serum cholesterol and numbers for LDL and HDL and a few other things too. So what do these letters represent? What's the biochemistry behind this? Well, the final L is a lipoprotein. And so what you have is the way the cell moves these water insoluble molecules around in your blood. What happens is that in those cells, they get put into a package where all the water is excluded, all the water repelling tails come to lie next to each other, and the polar head groups, to the extent that they have them, are on the outside. And in my cartoon here, you know, this contains fats, which has the 3 fatty acid chains, some cholesterol. This was some French publication, which is why it has this spelling of protein, and then just a cartoon version of a protein. And so you have a mixture of basically a lipid droplet, which has embedded in it and bound to its surface proteins that help facilitate and regulate the size of the droplet. And they also allow this droplet to be recognized by signals in the amino acid sequence on the surface of these proteins as to what is to be done with this. Is it to go to adipose tissue? Is it to go to the liver? Is it to be used for energy, or is it to be used for storage? Now, when you pour some oil into a pot of water on the stove, you find that the oil comes to the top and it floats. That's how my fire demonstration worked. On the other hand, if you put it in a piece of hamburger, it sinks to the bottom. And that's because lipid is a very low density, a lower density, lighter than water in terms of volume, weight per volume. Protein, on the other hand, is higher density than water. And so if you have a lipid droplet that has only a small amount of protein in it, its overall density is going to be lower. And so it's going to be a low density combination of lipid and protein, lipid protein. And that's what LDL stands for, low density lipoprotein. On the other hand, if there's a lot more protein associated with a lipid droplet, then it's going to be a high density lipoprotein, HDL. And let me say, these are very specific proteins. There are certain proteins that are found in the LDL, certain proteins that are found in the HDL. It's not just random protein. They have a real specific structure and a real specific way to be recognized by the other parts of the body. And so to greatly overgeneralize, the problem with LDL, you can see, is that it's just a big drop of liquid and does not have very many places in here that can bind to water. Whereas HDL is a really more soluble form of this lipid droplet. And to reduce it to an extremis, you know, the cartoon version here is that if there's a lot of LDL, high LDL in your body, that can just result in these plaques forming within your blood vessels. Whereas if you have a large amount of HDL, that is a more efficient way to solubilize and move these lipid droplets around. And there are multiple forms of HDL, not all of which turned out to be good. There are multiple forms of LDL. So there's layers of complexity here. And I'm really not that well informed on the physiology of this, but this is the sort of gross generalization of what's going on here. Okay, so to finish up here, one of the most widely used drugs in the world, as I said, are these statins. And they have found to very effectively lower blood cholesterol and very effectively lower the incidence of cardiovascular disease. So how do they work? The question I often like asking students is, what is a drug anyway? People take these drugs. Do you have any idea what it is in biochemical terms? What's the function of a drug? What does a drug do? And the answer is that most drugs are inhibitors of enzymes. So a drug to a biochemist is almost some small molecule that will bind to the active site of a protein and stop it from working. All right, so when I talked about the synthesis of cholesterol being so complicated, in an early step in forming those 4 fused rings, you have this molecule called HMG-CoA reductase, or HMG-CoA, and the enzyme that it binds to is HMG-CoA reductase. It was found, looking at some natural products, that they had parts of the molecule that look very much like this intermediate in cholesterol synthesis. And these were developed as statin drugs because they indeed will bind to the enzyme that's making cholesterol. But unlike HMG-CoA itself, they won't let go of the enzyme. They will bind in that active site and effectively block the enzyme from further action. So the effect is to lower the rate of cholesterol synthesis. You don't want to turn it off completely. That's why they often, want to calibrate what's the most effective dose for you. But it comes in various slight modifications. I mean, you will see that these 2 drugs, for example, are the same on this end, but this part out here that's going to bind to other parts of the protein are slightly different. And so they may be slightly different in how tightly they bind to the enzyme. Now, this is such an important human health issue that this has been studied in great detail. And we know the exact enzyme, the exact structure of the enzyme that the drug binds. This is a model of the drug itself. Here is the active site. We know all the amino acids. And so a lot of effort has been put into finding the exact mechanism by which they work and what the most effective form of it is. And it's found that, people vary slightly in the exact sequence of their amino acids, et cetera. And so some of these will work better for some people. Other slight versions will work better for other people. That's why they come in these different forms. Okay, so finally here, I say these statins are widely used. Back when I could see you all, maybe I'll still do it. If I ask for raising hands of how many people are taking statins, I'll bet more than 50% of the people in this group are taking statins. I take a statin. I've been taking statin for many, many years. They are so widely prescribed because they really have been effective. When they were first found, it was the people who had the most serious types of cholesterol levels were the target population for the drug. So there's disease called familial, which simply means inherited hypercholesterolemia. If you see anything that's an emia, it means in the blood. So if you have septicemia, means you've got bacteria in your blood. And if you have hyper high cholesterol in your blood, that's hypercholesterolemia. So people who have this inherited disease, in some of the more extreme forms of it, are people who are dying from heart failure in their 20s. Often they just were making an excess of cholesterol, or they had a defect in some of the carriers, HDL or LDL, such that they really just couldn't move the cholesterol around properly in their bloodstream. And it was found that with these statins, whereas the non-treated population in the extreme, more than 60% of the people would die within 10 years of the diagnosis. With the treatment with the statins, they could reduce the death rate by 80 or 90%. A few people have suggested that everybody should be taking statins. That might be a little bit extreme, but they do seem to be an example of one of the more mortality reducing drugs of all of the similar kind of drugs that are out there. Very effective. All right, so next time I'm going to have my focus on the protein part of our diet and nitrogen-containing molecules, and we'll wrap up the course in lecture number 5, looking at protein-rich food. So with that, let me open this up to questions. There are often a few questions relating to essential fatty acids and statins, and let me remind you, I am not an MD, but I do have some opinions about the biochemistry. Okay, questions out there? Questions? You can just unmute yourself if you have a question. Anybody? Well, maybe not.
Well, wait. So there's really a lot in here, Barry. It's fantastic. So the lipoproteins, both the LDL and the HDL, are carriers?
Yes. There are ways to transport those lipids in the blood. And so those proteins will, as they're bumping along the cells, if they see a receptor, a cell that's a match, they will bind to them and they will trigger the take-up of that lipid droplet into some particular cells. So that's how they get sent. They get packaged and they have signals on the protein say, take these to the storage cells, take this to the fatty tissue. And that's how they end up in the right place.
And the HDL has many more of those proteins, so it sends better information. And the LDL it doesn't have those proteins, and so it's.
It's probably not so much the information, but that the LDL, because it has few proteins, most of the exposed non-water binding part of that lipid droplet is exposed and can bind to, among other things, other LDL molecules. And so you could start to get you want it being carried around in discrete packets, but if it starts to clump in your bloodstream, then you can start to get the formation of these plaques. Now, I should warn you that it's a lot more complicated than that, but that's kind of the underlying chemistry involved. It's the solubility issue. Now, people who have mutations may actually have changed the sequence, that says, take me to the adipocyte, and they may get delivered to the wrong place. And so people with these inherited propensities to cardiovascular failure are often more of an information defect than a solubility defect.
Great.
Roger.
Yeah, we hear, and I think it was actually on one of your slides, was in reference to it, that the omega-3 acids, I think, reduce inflammation or prevent inflammation in the body. Can you tell me how that works on a molecular level? What is going on there?
It's a really complex phenomenon. And you know, so first of all, it's not the omega-3s. It's something probably 10 steps down from what the omega-3 that you eat in your food is converted to some long chain fatty acid with double bonds at just the right places. And those become signaling molecules. So they're almost act like hormones in a way, in that their function is not to make anything or degrade anything, but to turn on and off certain physiological processes. And inflammation in itself is a very complex process. So it's really hard to simplify what's going on. But if, you know, I use the example, if you have a wound or an infection, and so you have a need to get blood cells to aggregate in there, you have to put some signal molecule in your blood that says, bring blood cells here. And these essential fatty acids are the precursors to these signaling molecules. One of them is called the prostaglandins. I don't know if you ever hear of that, but prostaglandins are involved in inflammation. Okay. Violetta.
Yes, you showed that there are several statins. How to choose the right one? Some insurance for cover some, others doesn't cover, and it's very difficult. I don't know why, how to choose them.
Yeah, and I really can't tell you anything about that. I mean, that gets into, you know, they've been tested in different clinical trials. I think it's not really understood. I mean, the other issue with all drugs is I said they work by binding to a protein and generally slowing it down or turning it off. But the issue with all drugs is that the only place they bind? And the answer is no. They bind tightly to some places and very weakly to other places. And so that's why these different varieties of statins might have different effects in different people, because the side effects of drugs are usually caused by them binding to a site that is very similar to their target site, but not exactly the right site. And so you're turning off something you don't want to turn off. So not a very satisfactory answer, but that I would not venture to suggest how you choose one statin over another. It's complicated. And let's see, Sophie, you had a question. She's still here. Okay. No, I guess not.
Yeah. I would like to know what happens when oil or fat goes rancid, and what it does to our body if we consume rancid fat.
Yeah, so generally the term rancid is referring to an oxidation, meaning the oxygen, air, and the oxygen dissolved in the water, being a very reactive molecule, will bind and form molecular bonds with things like the unsaturated sites in fatty acids. And so again, the chemistry that you can get is complicated. So it can bind at different places. It can promote the binding of additional atoms. And why it might be toxic, I really don't know the answer to that. And I don't know to what extent it is known, because you could get such a variety of different molecular compounds. It's kind of hard to sort out which ones might be acting on our physiology. Okay. All right. Now, all right, I want to, was there another question? I want to answer a question you didn't ask, but always gets asked at one point or another, and that is, what about organic food? And I'm always so reluctant to get into this because it's almost an emotional issue with many people. And it's one of these things that everybody knows that organic food is better than regular food. And I'm afraid the scientific establishment who studies this sort of thing just doesn't agree with that. Been reading a really interesting book recently called Resetting the Table by a man who's spent his whole career on food and agricultural policy. I thought it was one of the more informative and kind of rational views of it. So let me just say a bit about the whole organic food business. The reason I think a lot of scientists have trouble with accepting the idea of organic food is that the principle of it is really not supported by science. And the principle of organic food is that there is a difference between natural chemicals versus synthetic chemicals. You know, an example would be, I think a lot of people like organic food because they say it has no pesticides. Well, it's not that it doesn't have any pesticides. Organic food itself is loaded with pesticides, natural pesticides. And there are at least 100 different kinds of pesticides which organic farmers can put on foods. It's a question of whether they're natural ones or unnatural ones. And it all depends on how much and where it goes. So, you know, that basis of it is just not grounded in good science. The start of the whole organic food movement was actually not pesticides, it was about fertilizer. And that's the other key difference between conventional food and organic food, is that organic food is not to be used, is not to be grown with any synthetic fertilizer. It's an interesting problem now that most of the organic food you buy in the market is not coming from some small farm, it's coming from big industrial organic farming. And because there's more of it made now, and if you want to use manure on your crop, initially you wanted that manure to come from organic sources, but there's just not enough of it to go around. So most of those nitrogen atoms that go into most organic food are from chemically synthesized nitrogen. That's just, they come from manure that comes from feedlots. And then the other issue is, well, is it, if it's better for you, yes, okay, go for it. And there have been a lot of studies. One of the more frequently referred to is one that came from Stanford a few years back, in which they did an analysis of many other clinical trials, et cetera, that had been done. And there's just no strong supporting evidence that people who eat organic food are healthier, or that people who eat conventional food are sicker. Now, the reason I'm interested in this, and I'm kind of wading into this, is because one of the issues here is there are a lot of people whose diet is determined by how much money they have. And is it better to eat, and you're buying food for your kids, is it better to buy 3 organic apples or 4 conventional apples? And the science would say, buy the 4 apples. You know, buy as much fruit and veggies as you can. It's the amount that you can get in your diet that's more important. I mean, one thing that is generally clear about organic food is that it is more expensive. And on average, something between 30–35% more expensive. And that's because it takes more resources to make it, and it has a lower yield per acre. And so, you know, it all kind of comes down to, you know, one of the things that bothers me about it is that absolutism. It's such a black and white issue. You cannot, if you are an organic farmer, test some growing technique if it doesn't apply to the organic standards. So there's a big issue in modern food, and you know, here in Santa Cruz, certainly there's a big push for organic food. But as a biochemist, and I think I would be in the consensus for people who study this, the scientific underpinnings of it are not strong. So I hope you're still all going to come back for the final lecture here. All right. I was worried about losing all my friends, saying I'm not a big supporter of organic food. But questions? I'm willing to discuss.
I have a question on just what you said. So you've laid it out as a pretty black and white issue that the science does not support it. But I have to imagine that there are serious people with good scientific backgrounds on the other side of the question, which who would argue with you. What are the other arguments, the most persuasive arguments on the other side of what you've just described?
Well, yeah.
If it were that simple, we wouldn't have organic food around. There's got to be some rationale other than just uninformed information, uninformed people about pesticides, etc. There's got to be more to it than that. So maybe it's hard for you to articulate that, but because you don't believe in it. But what's your thought?
Well, I think probably the number one issue is always are there pesticides on this food or not? And I guess the answer is, looking at the literature, I don't find, and these surveys of all of the of all of the kind of trials don't find, really strong side to support the idea that there is a health difference. I think the reason it's so acceptable, it gets us into similar questions about, it's a fear of the pesticide more than anything else. I think it's interesting that the underlying issue of the fertilizer is kind of, I think, not so much important in most people's minds. It's the fear of the pesticides. But I'm not being able to give you a good answer, because you're looking for what are the strongest scientific arguments on the other side, and I don't know of them.
Is it better for the environment or the soil?
Okay, good question. Good question. And the answer is it's complicated. Let me just address two issues. One, for example, one of the big changes in agriculture in the last 10 years, and a positive one, is called no-till agriculture. So one of the biggest sources of greenhouse gases is cultivation of crops, something like 20 to 30%. You dig up the crop, you expose all of that organic molecules to the oxygen, and the bacteria and the fungi go wild, and they degrade it, and they produce CO2. All right. And you kind of lose that organics that you want in your soil. So farmers more and more are just not cultivating that, but that means somehow you've got to get rid of the weeds. And conventional people use weed killers, or you can burn it, although that's a bad environmental thing. The organic people are using plastic. And at first they could maybe recycle some of this plastic, but in most of the organic food in which they're doing it, they're really using hundreds of square miles of plastic to keep down the weeds. A second environment, so which is better? Okay, so a second issue is the fertilizer one. So the conventional agriculture is becoming so high tech that essentially all the fertilizer that's going on the farm is being sucked right up by the plant. So the use of fertilizer has gone way down, and the problem with environmental runoff is really been and can be really addressed. Now, if you use a manure, has an advantage, and it's more carbon in there, but you really can't control the rate at which it's degraded. And it's very difficult to control the dose, and you can get a lot of runoff. So is it better for the environment? It can be, but it takes some really intensive work. Plus, you can't just put raw manure on fields. You can actually poison your crops if you do that. You really have to compost it. And so, you know, one of the reasons why organic food is more expensive, because you're just burning more fuel in order to do all of the weed control and management you have to do to process it. So it's very much a mixed bag. All
Thank
right,
you.
so other questions? Okay, well, I think we will wrap it up there then. So I hope to see you all for lecture number 5. And as I said, we're going to be talking about proteins and protein